bienvenidos a este seminario. Bueno, ya veis que tenemos unos cuantos cambios, empezamos a emitir por streaming, empezamos a grabarlos y vamos a ver si la cosa funciona. Animaros a seguir dando seminarios, a pesar de que esto impone un poquito más de tener las cámaras aquí mirándote, y sabiendo que te va a ver quizás mucha más gente y va a quedar grabado para la posteridad, pero bueno, yo creo que es algo que puede ser bueno para nosotros y para el instituto. So I'm going to introduce our speaker, is Gaby Layton. She is from the University of Cape Town in South Africa. She has a degree bachelor's degree and master's degree in biological sciences, and she is now working on her PhD. And the PhD is about, well, this idea of the effects of the urbanization and the animals on the animals, and how the animals can adapt to these urban environments, and how they can be exposed to some threats like some contaminants. In fact, she's here doing some analysis of organic contaminants, maybe also some heavy metals. It depends on the amount of sample that we have, and to study the exposure of these animals that are living in general in an urban environment. Gaby, I'm going to give some highlights of her curriculum. She was awarded with the Robert May Prize of the British Ecological Society by a paper that was published in Methods in Ecology and Evolution. And this is a type of award for the early career researchers. And it was in 2016, so it was, you were doing then the master or?
Honors, my last year of my undergrad.
Your last year of, so very good. And you also had another grant to come here and to analyze, for the analysis of these samples of the World Wild Fund, I think.
Yes,
Table
Mountain Fund.
Yes. So, yes. So thank you, Gaby, for giving the seminar, and all for you.
Thanks, Rafa, for that great introduction. So good afternoon, everyone. Like Rafa says, I'll be presenting some of my PhD research on the effects of urbanization on the foraging ecology of Cape Town caracal and the kinds of trade-offs that they have to face at the urban edge. So I'm a student at the Institute for Communities and Wildlife in Africa, which is based at the University of Cape Town in South Africa, and I'm working here at IREC for the next 3 weeks or so. So for the first time, the majority of people in the world now live in urban areas, with 1.4 million people being added to cities each week. So unsurprisingly, urbanization is recognized as the principal threat to biodiversity globally through habitat loss and fragmentation. And urban wildlife research is a growing field. There's been about 10 times increase in publications over the last 40 years or so. But recent reviews have showed that there's some pretty strong biases in this research, both in terms of taxon, but also topic. And one of the strongest biases is actually a geographical bias. And these kind of biases making generalizations quite difficult. And so the small number of studies published in Asia, South America, and Africa are particularly worrisome, given that these are the areas, as you can see on this map, that are experiencing the greatest population growth and subsequent urbanization. And these areas contain numerous biodiversity hotspots. And urbanization presents many formidable novel threats to wildlife, introducing new species and physical barriers, but also things like diseases and pollutants. And this creates an evolutionarily novel environment at the urban edge, to which, somewhat paradoxically, some species actually adapt quite well. And importantly, dietary requirement and foraging ecology appear to be key to urban species success. So carnivores are really useful models to study the effects of urbanization, as their ecology is significantly altered by urban living. They can tell us about all of the threats at once. However, large carnivores like the puma and the leopard on the previous slides, they were usually the first to disappear in urban landscapes. And so this leads to a process of kind of mesopredator release in urban areas. And that can significantly alter ecosystem trophic dynamics. And therefore, the carnivores that are left behind are usually smaller, more diverse, more adaptable species, like these ones. And these species can act as really good sentinels for ecosystem health in these areas, as their high trophic status leaves them vulnerable to bioaccumulation of pollutants. So this is a really great diagram. It shows 4 scenarios where predators can actually benefit from anthropogenic niches. So they can benefit through the availability of novel prey, things that they wouldn't normally hunt. Increased prey density can actually reduce search costs in some cases. Human disturbance can actually make it easier to capture prey in some cases. And additionally, new environments can provide novel opportunities that lead to novel predation techniques. So while there may be increased or bolstered abundance of prey or resources at the urban edge, optimal foraging theory predicts that there's usually a risk-reward trade-off. So you're trying to maximize your energy acquisition while minimizing your mortality risks. And often risks in urban areas are actually not really detectable by the animal. So things like diseases and pollutants. And so they'll therefore prefer a suboptimal habitat. And then if the risks outweigh the benefits, then they can get caught in what's known as an ecological trap. And traps can actually be pretty common in rapidly changing landscapes, and they can drive local extinctions. So the focus of my research is carnivore adaptation in urban areas, and I'm focusing on the caracal. They're a really adaptable, medium-sized cat. They have a really wide distribution, as you can see here. They're found in many different changing landscapes, but little is known about their space use and ecology, particularly not in urban areas. And so my work is focused all the way down here on the Cape Peninsula. It includes the Table Mountain National Park and the city of Cape Town, which is in grey on the map here. And it's a city of around 430,000 people, and it's in the southernmost part of South Africa. So Cape Town is the oldest city in South Africa, but not everyone realizes how wild it still is in some places. And before the project started, we didn't really know very much about the wildlife in the Table Mountain National Park. However, we have since found a population of caracals still living around the city. And so this is a really great photo of one of our tad caracals, which was taken quite recently, right at the top of Table Mountain. So large predators like the Cape leopard and Cape lion have been absent on the Cape Peninsula for decades, making this caracal the de facto apex predator. And as South Africa's fastest growing city, Cape Town now has a really high or pretty high population density, but it's also located inside a biodiversity hotspot, making it a World Heritage Site. And there's about 250 square kilometers of available wildlife habitat. It's mainly made up by the Table Mountain National Park, but this is broken up in many places by a really hard urban edge. And then together with the coastal edge, this represents a pretty significant barrier to gene flow, both on and off the peninsula. Yeah. So the peninsula itself also has an urban gradient, so there are more natural areas as you move south. And so it represents kind of a natural laboratory to explore the effects of urbanization. And so my work forms part of a larger research effort, which is called the Urban Caracal Project. And I'll be able to make use of much of the data that's collected on this project, including GPS collar relocations. So as a major part of the project, caracals were captured and collared for several months at a time, and general capture protocols were followed. Blood and hair were taken, and body measurements were taken. And then the collar data was uploaded in near real time using this web interface. And so this is just an example here for this juvenile male for about 2 weeks of data, really close by to my university campus in Cape Town. So the campus is just there. So these are just some of the caracals that we captured on the project. At the top here we have some of the males, females in the middle, and some of the juveniles at the bottom. So you can see a pretty wide range of expressions and ages. So to better understand the risk-reward trade-offs caracals are facing in selecting foraging habitat, I want to investigate associations between environmental toxicants and an urban diet, and to try and quantify this risk. And so we're really interested in persistent organic pollutants, or POPs. They're pretty ubiquitous. They bioaccumulate, as I'm sure you all know, through the food web. So this is an example diagram of a food web for the greater Cape Town area, both around the city but also in further afield. And so we know that they're highly toxic with multiple detrimental health effects, and high levels of organochlorine, compound pesticides, things like DDT, and also polychlorinated biphenyls, or PCBs, are strongly associated with urban areas. And studies show that they do negatively affect wildlife in Europe and North America, and that they're strongly linked to diet, but there's very little work that's been done around wildlife in African cities. So on the peninsula, we found that the caracals have a pretty low average annual survival, particularly, you can see here, for juvenile males. And looking at the causes of death in the collared cats, we can see that disease and pollutants actually do come up as pretty important threats. And there's already strong evidence of DDT and PCBs affecting other local species, like the Cape clawless otter and the black harrier. And so the sheer quantity of pollutants in the peninsula ecosystem could produce compounding effects in individuals exposed to multiple compounds. So to better understand pathways of pollutant exposure, I'm looking at a detailed assessment of compositional diet to quantify the use of urban prey. So other urban species that tend to do well consume about 30% urban prey, and we know from previous studies that the caracal is a really varied diet. It's an opportunistic generalist. And so we can predict that the Cape Town caracal will consume probably more urban than wild prey. And urban prey here just includes introduced species like this domestic cat or the grey squirrel, but also native human-associated species like the guinea fowl or the Egyptian goose that tend to do really well in transformed areas. So in this study, we use a combination of two diet methods to overcome limitations of each. So firstly, we use GPS collar data downloaded, and then we run an algorithm which identifies GPS clusters. And these are just aggregations of locations. And then these GPS clusters were investigated using standardized protocols as soon as possible in the field to try and find prey remains. And secondly, scats were collected and the prey remains were identified. So we ground truthed almost 700 GPS clusters from 22 collared cats. We found almost 400 kills, which are the red points here, and over 650 scats, which are the blue points here. We found that diet was really varied, but it was actually dominated by a few species. So guinea fowl are really common, then Cape cormorant and Egyptian goose. And then for the scats, it was a little bit more biased towards mammals, a slightly different pattern, very high dominance of flay rat, which is the kind of wetland-associated rodent, then Cape cormorant, and then rock hyrax, which is a medium-sized mammal. Unexpectedly, we see a really small proportion of introduced and domestic species in the diet, only about 11% of kills and 7% of scat. And there seem to be some demographic differences as well. So we see adult individuals taking more urban prey. The urban prey is the orange section here. And males seem to take more urban kills than females. So to investigate the trade-offs the caracals are navigating across the urban gradient, I want to look at landscape features that caracal prefer when hunting. So the traditional framework for classifying urban species is avoiders, so usually large species like puma, then adapters like bobcats, and exploiters like raccoons. However, there may be some variability in population responses and also the potential for change in these responses. So think something like a puma actually adapting to an urban area. So this kind of framework has been updated to allow for a range of different responses, so a species can be fully contained within one category or can span multiple categories. So we're quite interested in using the GPS cluster data to explore where caracals actually fall on this spectrum. And so here are some of our findings so far. So this is still a work in progress. So we're just looking at the proportion of GPS clusters with kills in each land use type. And we see that only around 5% of kills are in urban areas, again in the orange section. So to investigate some preliminary patterns in habitat preference, we looked at GPS clusters with kills and then those without. And most kills, around 60%, were made within 500 meters of the urban edge, which suggests to us that caracals are selectively foraging significantly closer to the edge. And then we found that male kills were also significantly closer to the edge, and juvenile kills were also significantly closer. And here we define juveniles as less than 2 years old. So there appears to be some individual level variation influencing the selection of predation sites. So even feeding on this really small component of urban prey on the fringes of urban areas seems to be sufficient to expose caracals to toxicants. We've already found exposure to several different kinds of rodenticides in the caracal, so we know from the diet results that this exposure is probably not through targeted introduced rodents, but maybe from tertiary exposure by consuming other carnivores that do consume those poisoned rodents, so things like genets and mongoose and raptors and also snakes. So while I'm here at IREC, I aim to assess the occurrence and patterns of pollutant exposure. So specifically, we're looking at things like DDT, DDE, multiple PCBs, and hopefully PBDEs as well. And we want to assess further the sources of contamination, look for possible health effects, and just like in some of the studies on these species that have been looked at here before. So I'm in the process of analyzing my blood and adipose samples at the moment. And then once I've finished with these results, I can begin to look further at relationships between concentrations and habitat. So particularly things like proportion urban area and other home range characteristics, as well as diet composition and a few different health measures. So as a sentinel and umbrella species, getting insight into the risks like pollutants that these caracals face can give us an idea of the threats that are faced by other species on the Cape Peninsula as well. And so we hope that the findings on the caracal can inform the conservation agencies in the area on management plans for biodiversity conservation more broadly. So just in summary here, we're looking at a trade-off between food security and environmental toxicant exposure risk at the urban edge. We've discovered really limited use of urban prey species. We've also found that the caracal are hunting closer to the urban edge, particularly males and juveniles. And these occasional visits into urban areas can probably be quite costly with potential exposure to pesticides, and we've already seen pretty high levels of rodenticide exposure. So the analysis I'm undertaking, coupled with ongoing genetic analysis, may go some way to understanding if caracals, along with many other species, are getting caught in an ecological trap. So I'd really like to thank my supervisors, everyone who helped me from data collection through to analysis, and the project funders and partners which are listed here. And I'd also really like to thank Rafa Mateo and IREC for hosting me while I'm here. Cool, thank you as well for your attention. And if you can, please follow the project on social media. Thanks, that's it. Cool.
Okay. Thank you, Gabi.
Yep.
I think we have time for questions, and maybe
any
of
questions?
you have questions, those working on carnivores, thinking, predation.
Yeah.
Well, I have a question. Well, one of your supervisors is Laurel Series. She was working in California, and she found some relationship between the exposure to some contaminants, in particular the anticolon rodenticides, and the presence of some infectious or parasitic diseases. It was mentioned in the lynx. I don't know if it was also in the pumas.
I think it was mainly the bobcats that she was looking at. Yeah.
Do you have something similar? Do you expect something similar with caracal, or the situation is a bit different?
So we are looking at the presence of different diseases in the caracal. So it is possible to look at an association or a relationship between the presence of disease and the presence of rodenticides. And I think Laurel is very interested in looking at if there is any link, similar to her work on bobcats. In terms of mange specifically, we haven't seen the same type of mange in the caracals at all. So we'll probably focus on different diseases or different measures of health, yeah. Mm-hmm.
And what about the causes of death in the animals that you are finding?
Most of them are roadkill, so most of them have been hit by cars, so that's our main cause of death. But within the collared cats, we see that things like diseases and exposure to rodenticides come up as more important, because obviously with our roadkill data, it'll be more biased towards animals we can actually recover, and roadkill is easier to find, so yeah.
Okay, thank you.
Mm-hmm.
Questions? Por cierto, si alguien que está viendo el vídeo en streaming quiere hacer preguntas, puede hacerlo a través del chat y las hacemos llegar aquí a la...
Thank you, Gabi, for your very interesting
It's
talk.
a pleasure. Thanks for coming.
So you showed that there were differences in the use of caracals depending on the gender, on the sex.
Mm-hmm.
Why do you think there are those?
So the differences between sexes?
Yeah.
So I think that males have much larger home ranges, so they're probably more likely to traverse greater areas and then come into contact with urban areas more often. And whereas the females tend to be much more focused on a smaller area, and generally it seems like they're avoiding urban areas more. So this is all sorry. Like preliminary analysis, but we are looking at resource selection functions. I'll be doing it with my GPS cluster data, but Laurel is also doing it with all the movement data that we've collected. So we'll begin to look at patterns like preference and things like that more in detail. But yes, it does seem like the females are avoiding urban areas.
Okay. And I have another question too.
Yes.
You have studied all the possibilities of getting polluted with, for instance, rodenticides from the urban areas.
Getting what, sorry?
Getting contaminated
yes,
or exposed to that.
Yes.
Have you thought about the risks that caracals could bring to human populations getting into urban cities?
The risks that caracals pose to humans?
Yeah, like diseases or something, like they could bring diseases from wild areas to...
Okay.
Is there something going on at the moment for that?
I would say that risk is probably pretty low. Like I said, they're only really using the urban fringes. They're not really penetrating into the urban matrix. They certainly don't come into contact with people. They're pretty shy, elusive animals. If you see one, it'll run away. I mean, they're very shy. So in terms of disease transmission to humans, I think the risk is pretty low. Maybe the risk to pets is higher, so because they feed on domestic cats, they might transmit diseases to domestic cats or dogs, but I would be more worried about the risk the other way around. So, you know, pets and things that they're eating actually transferring diseases to them, to a wild population. Yeah.
Yeah, well, there's been some work also going on, like with Echinococcosis multilocularis, like that are a parasite of rodents.
Okay.
And I mean, in the places where I've been working in France and also in Canada, they were studying something like that, like the coyotes, for instance, bring the echinococcosis towards urban populations, so there is higher chances to be infected because of these coyotes, for instance. So well, maybe something to think about it. Like,
it's an interesting idea. Yeah, thanks.
Thank you for the presentation. Just wondering, one of the outcomes that you can get from POP exposure in the animals, it's, you know, sublethal effects, like kind of reproductive failure or something like that. Do you have some life history data on this population that you can compare maybe to others that are not so related to urbanization?
So we've focused our study mainly on the peninsula, but there is a gradient of urbanization. So I guess we could compare the individuals that are further south to the ones that are further north or have more urban areas in their home ranges. We could make that comparison. There are other studies on caracals, more in agricultural areas, like further north in South Africa as well. But I mean, we would have to try and get access to that data.
You haven't compiled those data so far?
Sorry?
Yes.
We haven't?
Do you have already compiled those data that you can establish already a comparison besides the potential exposure to contaminants?
You mean like health measures and things like that? Yes, so yeah.
Productivity, etc.
Productivity
and
in terms
so
of...
have you seen any difference with non-urban related populations?
So we haven't yet made that comparison, but it would be a really useful one to make, to have a non-urban population versus urban. But, you know, I mean, our efforts and funding were limited, so we've really focused just on the Cape Peninsula, and it was a lot of effort to get that data on its own. So, yeah, but yes, it's a good comparison to make. Any other questions?
Okay, thank you for your talk.
It's a pleasure.
Very interesting.
Mm-hmm.
I guess you are mostly studying accidental contamination, accidental contact with pollutants. But have you considered the human attitudes towards the species? Because I understand that some part of the diet is
domestic,
human pets. So humans probably see the caracal as a threat for the pets. And what about the deliberate
poisoning
exposure to, for example, here is a problem, toxic baits, but is there this problem there?
So in terms of conflict with caracal and human-wildlife conflict, on the peninsula, I would say it's probably less than other areas in South Africa. So the caracal is actually listed as a vermin species in South Africa. And in some places where there's really intensive sheep farming, they actually do like block hunts, like culls, where they go out and they can shoot anything. But on the peninsula, I think people don't actually realize that the caracals are there often. So it's not like a widely known issue. And I think a lot of people who like blame the caracals for their cats going missing are often sensationalizing the issue a little bit. I mean, we only found like maybe 2 to 3% of domestic cats in their diet. It really isn't a big thing. So I don't think there's any really deliberate poisoning, not on the peninsula, certainly in other areas of the country. But there's some poisoning, but they're mainly shooting them. So yeah, very little deliberate poisoning, I would say. But other forms of persecution. So we have found some poaching, like gin traps. One instance where one of our cats was caught in a gin trap. And then there's, because we have quite a lot of unemployed and disadvantaged people living in the peninsula as well, there's quite a lot of bushmeat hunting. So people go out and they set sort of indiscriminate traps, and so we found some cats that have been caught in those as well, but I wouldn't really say it's targeted at caracals specifically. So, yeah.
Thank you. Have you made any studies on population trends in your study area? Or is a population which is open with receiving immigrants from the outside or whatever?
Yes. So that's what's quite interesting about this study area, is that it's actually a closed population. They're isolated by, I showed a map of the study area, and there's the city, which basically closes off the peninsula from the rest of the country. So it is an isolated population. And there's been some preliminary genetics showing that they are actually genetically isolated, and that there is a pretty high level of inbreeding, sometimes sibling to sibling breeding. But in terms of a population study, because we've only sampled like maybe 30 cats, it's not the whole population, so it would be difficult to make general trends statements about the whole population. So yeah, it's just a subset.